Hi, my name is Matt Fisher. I'm a chemistry professor at St. Vincent College, and I want to give everyone a quick overview of the session that I'll be facilitating at the 2020 SENCER Summer Institute on SENCER, COVID-19, and the next decade of undergraduate STEM education. Really, what this session is going to be about is an opportunity for all of us to talk about how our experience of COVID-19 and what we've learned from that might change how we structure science courses for majors and non-majors. So the coronavirus pandemic has been a global issue for just about 7 months now. And in that time, roughly 13.5 million people have become infected. As you can see, the rates of infection continue to climb. And in terms of the situation in the US, as you can see, while the number of new cases decreased from April 1st to early June, since early June it has significantly increased. And there is growing fear that the virus may once again be out of control in the country. You can see the current hospitalizations are up as well. In the same period of time, these same 7 months, we have gone from knowing absolutely nothing about the pathogen involved in this pandemic to the following. We have the complete sequence of the viral genome. We have identified all of the proteins in it. In part, that was done through comparison with other coronavirus genomes. But not only do we have the genome, we have a number of different diagnostic tests to identify the virus. These were all available within the first couple of months of the pandemic. We have collected the sequences of several thousand genomes from viral samples around the world and been able to use those to develop an epidemiological record of how the virus is likely to have been transmitted in various parts of the globe and where infections were seeded from. We have high-resolution three-dimensional structures of critically important viral proteins, such as the spike protein bound to the ACE2 cell surface receptor on respiratory cells that it binds to and starts the process of the virus entering the cell. We have detailed structures of the RNA-dependent RNA polymerase for the coronavirus that is absolutely integral for it to be able to copy its own genome and produce progeny virus particles. We've also identified a number of cellular proteins that these viral proteins will interact with, and researchers have used that information and that knowledge to identify approved drugs, drugs that are in clinical trials, or molecules that are in preclinical research phases prior to going into clinical trials that have the potential to be used as treatments for COVID-19 infections by affecting the function of the cellular proteins that are necessary for the virus to use to complete its life cycle. In terms of vaccine development, a process that historically has taken at the shortest somewhere around 4 to 6 years and typically a decade or longer. Within 6 months, we had over 100 vaccine candidates being developed. And at the time that I'm recording this video in mid-July, the New England Journal of Medicine had just published the phase 1 clinical trials for the mRNA vaccine that has been developed by Moderna in collaboration with the National Institutes of Health. Phase 1 clinical trial results that look very promising in that the vaccine is safe and actually does generate an immune response in healthy individuals. And that vaccine is now moving on to phase 2 and phase 3 clinical trials. We've seen countless examples of scientists across the world collaborating with each other. We've seen an explosion in the willingness of scientists to share research results even before they're formally published through the use of preprint servers so that other research groups would be able to build on those results as well. This is a level of collaboration that many scientific leaders have pointed out is almost unparalleled in modern times. So how has this changed our understanding of science? Obviously, the speed of scientific progress and how fast We've been able to learn things about the coronavirus and develop potential vaccines and potential new treatments, as well as a better understanding of the pathophysiology of the disease, the collaboration in science that I've mentioned, but also the process of science. So we're all familiar with the tentative, provisional nature of scientific knowledge, but now, rather than that playing out in scientific conferences and hallway conversations and in peer-reviewed literature that no one outside of the scientific community reads, now it's being played out in public. A perfect example of that, the recommendations about wearing masks. Whereas now the recommendation is that everyone should wear a mask whenever they go out of their house, early guidance on face masks said the opposite. As CNN pointed out a month ago, why has the guidance on face masks changed since the pandemic started? Because we've learned more, and we've incorporated that knowledge into the recommendations that are being conveyed to the public. In addition, preprints, which were growing in importance, have now exploded in terms of their importance for COVID-19. They've also had an impact on peer review, so you can see the surge here in terms of the number of studies that are being posted to the major preprint servers. In the early part of the year, this is data from Nature in early May. Here's more recent numbers from the bioRxiv server. You can see they have almost 6,400 articles that have been posted. Time for review of submissions has also significantly shortened. I'm seeing manuscripts now that from time of submission to time of acceptance, it may be somewhere on the order of 2 to 3 weeks if it's a really critically important paper. But what we're seeing is that a lot of the news stories that are being reported are drawing from preprints as well as peer-reviewed literature and perhaps not making the distinction. And a very nice example of the impact that peer review can have on the way the conclusions are presented is the D614G mutation that is now widespread around the globe. The preprint suggested that the mutation had an impact on the severity of the infection, and it turns out that by the time the manuscript was peer-reviewed and published, the reviewers had challenged the authors on that, and the authors had to revise the manuscript appropriately. We also have questions about quality control in science. Again, things that we're all familiar with from our work in research laboratories, but that now are playing out in the public when we're making public policy recommendations and decisions. And so, for example, the COVID testing project based on the West Coast has been looking at a number of the tests that have been recently developed to look for antibodies against the SARS-CoV-2 virus. Some of those antibody tests are quite good. Others are very problematic in terms of the rate of false positives that they give and the number of real positives that they actually miss. And then finally, unlike major pandemics in the past, we have to deal with the impact of misinformation. In fact, this infodemic is something that the World Health Organization has identified as a public health crisis, in some ways as important, as critical as the actual COVID-19 pandemic. So how do we help our students understand not only how they can evaluate information, but how do they help friends and family members understand what is valid information versus what is misinformation? Finally, in the context of the United States and the ongoing demonstrations and debates about systemic racism since the murder of George Floyd, COVID-19 has highlighted, again, racial inequity in the U.S. when you look at the number of coronavirus cases per 10,000 people for all individuals and then compare that white, black, Latino. Black and Latino communities are seeing 3 times the rate of coronavirus cases. And you can see that the numbers across all age ranges are significantly higher for Black and Latino communities. than they are for white. So this is a virus that is not affecting all racial groups in the United States equally. Nationwide, black people are dying at a rate 2.5 times higher than that of white people. So here we have a public health pandemic that is not only had an impact on our understanding of the nature of science and the way that the general public views that, but is also having significantly different impacts on different groups within our society. So how might all of this impact STEM education, both for non-science majors and for STEM majors? And my question is not about how did it impact education in the spring 2020 semester, nor about how it might impact STEM education in the next year. Those are both important questions, but those are different than the one that my session will focus around, which will be what's the impact of our experience living through the COVID-19 pandemic on the way that we construct learning environments for our students, on what we ask our students to do, on what we expect them to learn for the next 5 to 10 years. SENCER had its origins in the AIDS epidemic, in Monica Vanis's course at Rutgers that taught through HIV and AIDS to some fundamental scientific concepts that Monica hoped would empower students to be able to act appropriately in that time of great uncertainty. What opportunities does the COVID-19 pandemic offer the SENCER community in terms of how we think about and the type of courses for STEM education that we create? That's what this session will focus on. Please bring your ideas. Please bring your questions. Please bring your concerns. Please bring what you're working on so that we can learn from each other and together outline how this pandemic might impact the next generation of students.